Proteins are crucial macronutrients for insects, playing diverse and essential roles in growth, development, reproduction, metabolism, immune function, and overall physiological health. Here's a detailed overview of the importance of proteins for insects. Structural components. Proteins serve as structural components in insect tissues, providing strength, flexibility, and support to cells, muscles, organs, and the cuticle. Structural proteins such as collagen, actin, myosin, and tubulin form the cytoskeleton, muscle fibers, and other cellular structures. Cuticular proteins, including chitin-binding proteins and structural cuticular proteins, contribute to the formation and remodeling of the insect cuticle, exoskeleton, and integument. Growth and development. Proteins are essential for growth, development, and tissue repair in insects. Amino acids, the building blocks of proteins, are required for synthesizing new proteins, enzymes, hormones, and structural components during larval growth, molting, metamorphosis, and adult tissue remodeling. Adequate protein intake is necessary for larval development, pupation, eclosion, and adult maturation in insects. Enzymes and metabolic pathways. Proteins act as enzymes, catalyzing biochemical reactions and metabolic pathways essential for energy production, nutrient metabolism, and cellular homeostasis. Enzymes such as proteases, lipases, carbohydrases, and nucleases facilitate the digestion, absorption, and utilization of dietary nutrients. Metabolic enzymes such as kinases, dehydrogenases, and synthetases regulate metabolic processes such as glycolysis, the citric acid cycle, and oxidative phosphorylation. Hormones and signaling molecules. Proteins serve as hormones, neurotransmitters, and signaling molecules that regulate physiological processes, behavior, and development in insects. Hormonal proteins such as juvenile hormones, JH ecdysteroids, insulin-like peptides, ILPs, and neuropeptides coordinate growth, metamorphosis, reproduction, and stress responses. Signaling proteins such as receptors, ligands, and transcription factors mediate cellular responses to environmental cues, hormonal signals, and metabolic changes. Immune function. Proteins play crucial roles in the insect immune system, defending against microbial pathogens, parasites, and environmental stresses. Immune proteins such as antimicrobial peptides, AMPs, lysozymes, lectins, and phenyloxidases act as effectors of innate immunity, disrupting microbial cell membranes, inhibiting pathogen growth, and facilitating phagocytosis and encapsulation by hemocytes. Immune recognition proteins such as pattern recognition receptors, PRRs, and complement-like proteins detect and respond to microbial invaders, activating immune responses and promoting host defense mechanisms. Reproductive health. Proteins are essential for reproductive processes in insects, including gametogenesis, fertilization, embryonic development, and egg production. In males, proteins are necessary for spermatogenesis, sperm maturation, and reproductive behavior. In females, proteins are required for oogenesis, eggshell formation, vitellogenesis, and oviposition. Proteins such as yolk proteins, chorion proteins, and vitellin play roles in egg development, nutrient provisioning, and embryonic viability. Behavioral responses. Proteins influence insect behavior, feeding preferences, foraging strategies, and communication signals. Behavioral proteins such as odorant-binding proteins, OBPs, pheromone-binding proteins, PBPs, and gustatory receptors, GRs, detect environmental cues, chemical signals, and food odors, guiding insect behavior such as host seeking, mating, and oviposition. Social insects use pheromones, cuticular hydrocarbons, and colony-specific proteins for communication, colony organization, and division of labor. In summary, proteins are essential macronutrients for insects, serving as structural components, metabolic catalysts, signaling molecules, immune effectors, and reproductive factors. Understanding the importance of proteins for insects provides insights into their nutritional ecology, physiological adaptations, and ecological interactions with their environment. Fatty acids, ChatGPT. Fatty acids are important components of insect nutrition, serving as essential constituents of cell membranes, energy storage molecules, signaling molecules, and precursors for various metabolic pathways. Here's a detailed overview of the importance of fatty acids for insects. Structural components. Fatty acids are major components of phospholipids, glycolipids, and cholesterol esters that comprise cell membranes and organelle membranes in insect cells. Fatty acids contribute to membrane fluidity, integrity, and permeability, influencing cellular processes such as ion transport, receptor signaling, and membrane trafficking. Saturated fatty acids, unsaturated fatty acids, and polyunsaturated fatty acids have distinct effects on membrane properties and cellular function. Energy storage. Fatty acids serve as energy storage molecules in the form of triglycerides, which are stored in specialized fat body tissues and lipid droplets throughout the insect body. During periods of energy surplus, excess dietary carbohydrates and proteins are converted into fatty acids through lipogenesis and esterification reactions. Fatty acids are stored as triglycerides in lipid droplets, providing a source of metabolic fuel for energy production during periods of fasting, flight, or increased energy demand. Metabolic fuel. Fatty acids are oxidized in mitochondria through beta-oxidation pathways to generate acetyl-CoA molecules, which enter the citric acid cycle Krebs cycle for ATP production through oxidative phosphorylation. Fatty acid oxidation yields more ATP per molecule compared to glucose metabolism, making fatty acids an efficient energy source for prolonged periods of activity, such as flight in insects. Insect flight muscles rely heavily on fatty acid oxidation for sustained flight performance and endurance. Lipid signaling. Fatty acids serve as signaling molecules and regulators of metabolic pathways, gene expression, and physiological processes in insects. Lipid-derived signaling molecules such as prostaglandins, eicosanoids, and oxylipins modulate immune responses, inflammation, reproduction, and behavior. Fatty acid metabolism and signaling pathways are regulated by enzymes such as lipases, fatty acid synthases, and desaturoses, which control the synthesis, modification, and degradation of fatty acids. Cuticular lipids. Fatty acids contribute to the composition of cuticular lipids, waxes, and hydrocarbons that form the insect cuticle, exoskeleton, and integument. Cuticular lipids provide waterproofing, protection against desiccation, and chemical defense against environmental stresses such as pathogens, predators, and ultraviolet UV radiation. Fatty acids are synthesized by epidermal cells, secreted onto the cuticle surface, and modified by enzymes such as elongases, desaturoses, and hydroxylases. Reproductive health. Fatty acids are important for reproductive processes in insects, including gametogenesis, sperm maturation, oogenesis, vitellogenesis, and egg production. Lipid-rich yolk proteins, lipoproteins, and phospholipids are synthesized in the fat body and transported to developing oocytes for egg formation and embryonic development. Fatty acid composition influences the nutritional quality, viability, and developmental success of eggs and offspring. Environmental adaptations. Fatty acid composition can vary among insect species and populations in response to environmental factors such as temperature, humidity, diet, and habitat. Insects may adjust their fatty acid profiles through diet selection, lipid metabolism, and cuticular lipid synthesis to cope with environmental stresses, thermal regulation, and physiological demands. Changes in fatty acid composition can influence membrane fluidity, thermal tolerance, and cold hardiness in insects. In summary, fatty acids are essential components of insect nutrition, serving as structural components, energy storage molecules, metabolic fuels, signaling molecules, cuticular lipids, and reproductive factors. Understanding the importance of fatty acids for insects provides insights into their nutritional ecology, physiological adaptations, and ecological interactions with their environment. Sterols, ChatGPT. Sterols are important lipid molecules in insect physiology, playing crucial roles in membrane structure, hormone synthesis, growth, development, reproduction, and other physiological processes. Here's a detailed overview of the importance of sterols for insects. Membrane structure. Sterols, particularly cholesterol and ergosterol, are major components of cell membranes and organelle membranes in insect cells. Sterols help maintain membrane integrity, stability, and fluidity by interacting with phospholipids and regulating membrane dynamics. Sterol-enriched lipid wraps and microdomains play roles in signal transduction, receptor trafficking, and membrane protein organization in insect cells. Hormone synthesis. Sterols serve as precursors for the synthesis of steroid hormones, including ecdysteroids, molting hormones, and juvenile hormones JH, which regulate growth, development, metamorphosis, and reproduction in insects. Ecdysteroids such as ecdysone and 20-hydroxyecdysone are synthesized from cholesterol in the prothoracic glands and regulate molting, metamorphosis, and developmental transitions. Juvenile hormones such as JH3 are synthesized from cholesterol in the corpora allata and regulate larval growth, metamorphosis, and reproductive maturation. Growth and development. Sterols are essential for growth, development, and tissue differentiation in insects. Sterol biosynthesis and metabolism are tightly regulated during larval growth, molting, and metamorphosis to ensure proper tissue remodeling, cuticle formation, and organ development. Sterol deficiency or inhibition of sterol biosynthesis can disrupt developmental processes, leading to developmental abnormalities, growth arrest, or lethality in insects. Reproductive health. Sterols play roles in reproductive processes, including gametogenesis, spermatogenesis, oogenesis, vitellogenesis, and egg production in insects. Sterol-rich lipoproteins, such as high-density lipoproteins (HDLs) and low-density lipoproteins (LDLs), transport sterols and lipids to developing oocytes for egg formation and embryonic development. Steroid hormones regulate reproductive behaviors, mating success, and sexual differentiation in males and females. Lipid metabolism. Sterols are involved in lipid metabolism, including cholesterol synthesis, uptake, transport, and storage in insect tissues. Sterol regulatory element binding proteins (SREBPs) and other transcription factors regulate the expression of genes involved in sterol biosynthesis, uptake, and homeostasis in response to nutritional cues and metabolic demands. Sterol esterification and storage in lipid droplets contribute to energy reserves and metabolic flexibility in insects. Environmental adaptations. Sterol metabolism and composition can vary among insect species and populations in response to environmental factors such as temperature, humidity, diet, and habitat. Insects may adjust their sterol profiles through dietary selection, sterol synthesis, and sterol binding proteins to cope with environmental stresses, thermal regulation, and physiological demands. Changes in sterol composition can influence membrane properties, fluidity, and permeability in response to environmental cues. Cuticular lipids. Sterols contribute to the composition of cuticular lipids, waxes, and hydrocarbons that form the insect cuticle, exoskeleton, and integument. Sterols provide waterproofing, flexibility, and structural support to the cuticle, protecting insects against desiccation, mechanical damage, and environmental stresses. Sterol-enriched epicuticular lipids and wax layers help reduce water loss, repel pathogens, and maintain integumental integrity in insects. In summary, sterols are essential lipid molecules for insects, serving as structural components, hormone precursors, signaling molecules, reproductive factors, and metabolic regulators. Understanding the importance of sterols for insects provides insights into their physiological adaptations, developmental processes, reproductive strategies, and ecological interactions with their environment. Nucleic acids, ChatGPT. Nucleic acids are essential biomolecules for insects, serving as the carriers of genetic information DNA and RNA, and playing critical roles in gene expression, protein synthesis, growth, development, reproduction, and cellular function. Here's a detailed overview of the importance of nucleic acids for insects. Genetic material. Nucleic acids, particularly DNA, deoxyribonucleic acid, contain the genetic instructions that determine the structure, function, and behavior of organisms. Insects possess nuclear DNA, which is organized into chromosomes within the cell nucleus, and mitochondrial DNA, which is found in mitochondria and encodes genes essential for energy production and metabolism. DNA sequences encode genes, regulatory elements, and non-coding regions that control developmental processes, physiological functions, and adaptive traits in insects. Gene expression. Nucleic acids regulate gene expression through transcription, mRNA processing, and translation processes. DNA serves as a template for the synthesis of messenger RNA, mRNA molecules, which carry genetic information from the nucleus to the cytoplasm for protein synthesis. RNA polymerase enzymes transcribe DNA into pre-mRNA transcripts, which are modified through splicing, capping, and polyadenylation to produce mature mRNA molecules. mRNA molecules are translated by ribosomes into proteins, which perform diverse functions in insect cells, tissues, and organs. Protein synthesis. Nucleic acids, specifically RNA, ribonucleic acid, play central roles in protein synthesis and ribosome function. Transfer RNA turn molecules decode mRNA codons and deliver amino acids to ribosomes during translation, ensuring the accurate and efficient synthesis of polypeptide chains. Ribosomal RNA, rRNA, molecules constitute the structural and catalytic components of ribosomes, which catalyze peptide bond formation and coordinate the assembly of amino acids into proteins. Messenger RNA, mRNA, molecules carry genetic information from the nucleus to the cytoplasm and serve as templates for protein synthesis by ribosomes. Regulatory elements. Nucleic acids contain regulatory elements, such as promoters, enhancers, silencers, and transcription factor binding sites, that control gene expression and cellular processes in response to developmental cues, environmental signals, and physiological states. DNA sequences regulate the timing, magnitude, and spatial distribution of gene expression during embryonic development, tissue differentiation, and physiological responses in insects. Epigenetic modifications, such as DNA methylation and histone modifications, influence chromatin structure, gene accessibility, and transcriptional activity in insect genomes. Genetic variation. Nucleic acids contribute to genetic variation and evolutionary diversity in insect populations through processes such as mutation, recombination, and gene flow. DNA mutations, including base substitutions, insertions, deletions, and chromosomal rearrangements, generate genetic diversity and allelic variation within insect populations. Recombination events during meiosis shuffle genetic material between homologous chromosomes, creating new combinations of alleles and promoting genetic adaptation to changing environments. Gene flow between populations introduces genetic variation and facilitates the spread of advantageous traits in insect species. Developmental processes. Nucleic acids regulate developmental processes such as embryogenesis, metamorphosis, and tissue differentiation in insects by controlling the expression of developmental genes and signaling pathways. Transcription factors, signaling molecules, and regulatory RNAs coordinate cell fate determination, pattern formation, and morphogenic events during embryonic development and organogenesis. Post-transcriptional regulatory mechanisms, such as microRNA and small interfering RNA (siRNA) pathways, fine-tune gene expression and developmental processes in response to internal and external cues. Reproductive biology. Nucleic acids play crucial roles in reproductive biology, including gametogenesis, fertilization, embryonic development, and parental care behaviors in insects. Meiotic divisions during gametogenesis produce haploid gametes, sperm and eggs with unique combinations of genetic material. Fertilization of eggs by sperm initiates embryonic development, zygotic genome activation, and morphogenic processes leading to embryo formation and organogenesis. Maternal RNAs and proteins deposited in eggs contribute to early embryonic development and provide maternal provisioning for offspring viability and fitness. In summary, nucleic acids are fundamental biomolecules for insects, serving as carriers of genetic information, regulators of gene expression, mediators of protein synthesis, drivers of developmental processes, contributors to genetic variation, and facilitators of reproductive biology. Understanding the importance of nucleic acids for insects provides insights into their genetic diversity, evolutionary adaptations, developmental plasticity, and ecological interactions with their environment. Inorganic salts. ChatGPT. Inorganic salts, also known as mineral salts or electrolytes, are essential nutrients for insects, contributing to various physiological processes, osmoregulation, pH balance, nerve function, muscle contraction, and enzyme activity. Here's a detailed overview of the importance of inorganic salts for insects. Osmoregulation. Inorganic salts play crucial roles in osmoregulation, helping insects maintain internal ion balance, osmotic pressure, and water balance in their body fluids and tissues. Sodium, Na plus potassium, K plus chloride, Cl calcium, Ca2 plus magnesium, Mg2 plus, and other ions are actively transported across cell membranes by ion channels, pumps, and cotransporters to regulate osmotic gradients and cellular hydration. Osmoregulatory organs such as Malpighian tubules and rectal glands facilitate ion transport, water reabsorption, and excretion of nitrogenous wastes to maintain osmotic homeostasis in insects. pH regulation. Inorganic salts contribute to pH regulation and acid-base balance in insect hemolymph, circulatory fluid, and tissues. Bicarbonate ions HCO3 and carbonate ions CO32 act as buffering agents, neutralizing acids and bases to maintain physiological pH levels. Acid-base balance is essential for enzyme activity, protein function, ion transport, and metabolic processes in insects. Insects regulate pH through respiratory gas exchange, ion transport, and excretory mechanisms to prevent acidosis or alkalosis. Nerve function. Inorganic salts are essential for nerve function, neurotransmission, and neuronal excitability in insect nervous systems. Sodium, Na plus, and potassium, K plus ions play critical roles in generating action potentials, propagating nerve impulses, and depolarizing and repolarizing neuronal membranes. Ion channels such as voltage-gated sodium channels and potassium channels regulate ion fluxes and membrane potential changes, allowing for rapid and coordinated communication between neurons and sensory organs. Muscle contraction. Inorganic salts are necessary for muscle contraction, movement, and locomotion in insects. Calcium ions Ca2 plus bind to troponin and trigger muscle contraction by releasing stored calcium from the sarcoplasmic reticulum and activating the contractile proteins actin and myosin. Magnesium ions Mg2 plus modulate the activity of ATP-dependent myosin ATPase, regulating cross-bridge cycling and muscle relaxation. Sodium, Na plus, and potassium, K plus ions maintain membrane excitability and action potential propagation in muscle fibers. Enzyme activity. Inorganic salts serve as cofactors, activators, or inhibitors for enzymes involved in various metabolic pathways, biochemical reactions, and cellular processes in insects. Metal ions such as magnesium Mg2 plus, zinc Zn2 plus, iron Fe2 plus, Fe3 plus, copper Cu2 plus, and manganese Mn2 plus coordinate enzyme structure and catalytic activity, facilitating substrate binding, chemical reactions, and product formation. Metalloenzymes, such as metalloproteases, metallohydrolases, and metallooxidases, require metal ions for their function and stability. Water balance. Inorganic salts influence water balance, fluid distribution, and hemolymph volume in insect circulatory systems. Sodium ions, Na plus, and chloride ions, Cl, contribute to hemolymph osmolality and ion concentration gradients, driving water movement between compartments and tissues. Osmotic gradients created by inorganic salts facilitate water absorption in the digestive tract, water reabsorption in the excretory system, and water retention in cells and tissues to prevent dehydration and maintain hydration. Metabolic regulation. Inorganic salts regulate metabolic pathways, enzyme kinetics, and metabolic fluxes in insect cells, tissues, and organs. Phosphate ions PO43 serve as essential components of ATP, nucleic acids, and phosphorylated intermediates in energy metabolism, biosynthesis, and signal transduction pathways. Inorganic ions such as sulfate ions, SO42, and nitrate ions number 3 participate in sulfur metabolism, nitrogen metabolism, and amino acid synthesis in insects. In summary, inorganic salts are essential nutrients for insects, contributing to osmoregulation, pH balance, nerve function, muscle contraction, adaptation. Understanding the importance of inorganic salts for insects provides insights into their physiological adaptations, ecological interactions, and nutritional requirements in diverse environments. Microorganisms. ChatGPT. Microorganisms play significant roles in the ecology, physiology, and health of insects, influencing nutrient acquisition, digestion, symbiosis, immune function, and environmental adaptation. Here's a detailed overview of the importance of microorganisms for insects. Nutrient acquisition and digestion. Microorganisms aid in the digestion and utilization of complex nutrients in insect diets, including cellulose, lignin, proteins, and lipids. Symbiotic microorganisms such as bacteria, fungi, and protozoa produce enzymes such as cellulases, proteases, lipases, and amylases that break down plant cell walls, proteins, and carbohydrates into simpler molecules that insects can absorb and utilize. Gut microbiota enhance nutrient extraction, energy metabolism, and digestive efficiency in herbivorous, detritivores, and saprophagus insects. Symbiotic relationships. Insects form symbiotic relationships with diverse microorganisms, including mutualistic, commensalistic, and parasitic associations, which provide benefits such as nutrition, defense, detoxification, and reproduction. Mutualistic symbionts such as Buchnera bacteria in aphids, Lochmannia bacteria in ants, and Wolbachia bacteria in various insect species provide essential nutrients. Microorganisms play significant roles in the ecology, physiology, and health of insects, influencing nutrient acquisition, digestion, symbiosis, immune function, and environmental adaptation. Here's a detailed overview of the importance of microorganisms for insects. Nutrient acquisition and digestion. Microorganisms aid in the digestion and utilization of complex nutrients in insect diets, including cellulose, lignin, proteins, and lipids. Symbiotic microorganisms such as bacteria, fungi, and protozoa produce enzymes such as cellulases, proteases, lipases, and amylases that break down plant cell walls, proteins, and carbohydrates into simpler molecules that insects can absorb and utilize. Gut microbiota enhance nutrient extraction, energy metabolism, and digestive efficiency in herbivorous, detritivores, and saprophagus insects. Symbiotic relationships. Insects form symbiotic relationships with diverse microorganisms, including mutualistic, commensalistic, and parasitic associations, which provide benefits such as nutrition, defense, detoxification, and reproduction. Mutualistic symbionts such as Buchnera bacteria in aphids, Lochmannia bacteria in ants, and Wolbachia bacteria in various insect species provide essential nutrients such as amino acids, vitamins, and cofactors that complement the insect's diet. Commensal microorganisms inhabit insect guts, cuticles, or reproductive organs without causing harm or benefit. Parasitic microorganisms such as pathogens, parasites, and parasitoids can cause diseases, infections, or mortality in insect populations, influencing population dynamics, community structure, and ecosystem function. Immune function. Microorganisms interact with the insect immune system, modulating immune responses, microbial recognition, and host defense mechanisms against pathogens, parasites, and environmental stresses. Gut microbiota stimulate the development and activation of the insect immune system, promoting the production of antimicrobial peptides, AMPs, reactive oxygen species, ROS, and phagocytic hemocytes that combat microbial invaders. Microbial symbionts produce antimicrobial compounds, compete for resources, and modulate host immunity to maintain symbiotic homeostasis and prevent pathogen colonization. Environmental adaptation. Microorganisms facilitate environmental adaptation and niche colonization in diverse habitats, including soil, water, plants, and animal hosts. Insects harbor microbial communities that are specialized for specific ecological niches, feeding habits, and microhabitats, reflecting adaptations to local environmental conditions, dietary preferences, and host associations. Gut microbiota composition can vary among insect species, populations, and developmental stages in response to environmental factors such as diet quality, habitat structure, microbial interactions, and biotic interactions. Reproductive biology. Microorganisms influence reproductive biology, mating behavior, fertility, and offspring fitness in insects through mechanisms such as pheromone production, mate recognition, and reproductive organ development. Microbial symbionts can modulate sex pheromone production, mate choice, and reproductive success in insects, affecting mating behavior and sexual selection. Maternal transmission of symbiotic microorganisms from parent to offspring can shape offspring microbiota composition, developmental trajectories, and fitness-related traits such as growth, survival, and reproductive performance. Biological control. Certain microorganisms have been utilized for biological control of insect pests in agriculture, forestry, and public health, offering environmentally friendly alternatives to chemical pesticides. Entomopathogenic microorganisms such as bacteria, e.g., Bacillus thuringiensis fungi, e.g., Bovaria bassiana viruses, e.g., nucleopolyhedroviruses, and nematodes, e.g., Steiner macarpocapsae infect and kill pest insects, reducing pest populations and crop damage. Microbial biopesticides are selectively toxic to target pests, minimally harmful to non-target organisms, and compatible with integrated pest management (IPM) strategies. Ecological interactions. Microorganisms participate in complex ecological interactions and trophic relationships within insect communities, influencing food webs, nutrient cycling, and ecosystem dynamics. Microbial decomposers and detritivores break down organic matter, recycling nutrients and energy in terrestrial and aquatic ecosystems. Microbial symbionts mediate interactions between herbivores, predators, parasites, and pathogens, shaping insect population dynamics, community structure, and biodiversity. Microbial diversity and ecosystem function are interconnected, with microorganisms playing key roles in ecosystem services such as pollination, nutrient cycling, and soil fertility. In summary, microorganisms are integral components of insect biology, ecology, and evolution, influencing nutrient acquisition, digestion, symbiosis, immune function, environmental adaptation, reproductive biology, biological control, and ecological interactions. Understanding the importance of microorganisms for insects provides insights into their nutritional ecology, physiological adaptations, host-microbe interactions, and ecological roles in diverse ecosystems. Nutrition and growth. ChatGPT. Nutrition is critically important for the growth and development of insects, as it provides the necessary energy, building blocks, and regulatory signals for various physiological processes. Here's how nutrition influences growth in insects. Energy provision. Nutrition provides insects with the energy needed for growth, metabolism, and physiological activities. Carbohydrates, lipids, and proteins obtained from the diet are metabolized through cellular respiration to generate ATP, the energy currency of cells. Adequate energy intake ensures optimal metabolic rates and energy balance, supporting growth processes such as cell division, tissue differentiation, and organ development. Structural components. Nutrition supplies insects with the raw materials necessary for building and maintaining structural components of the body, including proteins, lipids, carbohydrates, and minerals. Amino acids from dietary proteins are used to synthesize structural proteins, enzymes, hormones, and other functional molecules required for growth and development. Lipids contribute to the formation of cell membranes, neural tissues, and energy reserves. Carbohydrates serve as energy sources and precursors for chitin synthesis in the insect cuticle and exoskeleton. Hormonal regulation. Nutrition influences hormonal signaling pathways that regulate growth, molting, metamorphosis, and reproductive development in insects. Hormones such as icossteroids, molting hormones, and juvenile hormones (JH) coordinate developmental transitions and physiological responses to nutritional cues. Nutrient availability, dietary quality, and energy balance can modulate hormone synthesis, secretion, and sensitivity, affecting growth rates, developmental timing, and body size in insects. Molting and metamorphosis. Nutrition plays a crucial role in the molting process and metamorphic transformations that occur during insect development. Molting is a coordinated sequence of physiological events involving the synthesis and degradation of the old cuticle, resorption of nutrients, and formation of a new cuticle. Adequate nutrition, particularly protein and chitin, is essential for producing a flexible and expandable exoskeleton that can accommodate growth during each molting cycle. Metamorphosis, such as complete metamorphosis, e.g., egg, larva. Nutrition and growth, ChatGPT. Nutrition is critically important for the growth and development of insects, as it provides the necessary energy, building blocks, and regulatory signals for various physiological processes. Here's how nutrition influences growth in insects. Energy provision. Nutrition provides insects with the energy needed for growth, metabolism, and physiological activities. Carbohydrates, lipids, and proteins obtained from the diet are metabolized through cellular respiration to generate ATP, the energy currency of cells. Adequate energy intake ensures optimal metabolic rates and energy balance, supporting growth processes such as cell division, tissue differentiation, and organ development. Structural components. Nutrition supplies insects with the raw materials necessary for building and maintaining structural components of the body, including proteins, lipids, carbohydrates, and minerals. Amino acids from dietary proteins are used to synthesize structural proteins, enzymes, hormones, and other functional molecules required for growth and development. Lipids contribute to the formation of cell membranes, neural tissues, and energy reserves. Carbohydrates serve as energy sources and precursors for chitin synthesis in the insect cuticle and exoskeleton. Hormonal regulation. Nutrition influences hormonal signaling pathways that regulate growth, molting, metamorphosis, and reproductive development in insects. Hormones such as icossteroids, molting hormones, and juvenile hormones (JH) coordinate developmental transitions and physiological responses to nutritional cues. Nutrient availability, dietary quality, and energy balance can modulate hormone synthesis, secretion, and sensitivity, affecting growth rates, developmental timing, and body size in insects. Molting and metamorphosis. Nutrition plays a crucial role in the molting process and metamorphic transformations that occur during insect development. Molting is a coordinated sequence of physiological events involving the synthesis and degradation of the old cuticle, resorption of nutrients, and formation of a new cuticle. Adequate nutrition, particularly protein and chitin, is essential for producing a flexible and expandable exoskeleton that can accommodate growth during each molting cycle. Metamorphosis, such as complete metamorphosis, e.g., egg, larva, pupa, adult, or incomplete metamorphosis, e.g., egg, nymph, adult, requires precise coordination of hormonal signals, nutritional resources, and physiological changes to ensure successful transition between life stages. Developmental plasticity. Nutrition influences the developmental plasticity of insects, allowing them to adjust their growth trajectory, life history strategies, and phenotypic traits in response to environmental conditions, resource availability, and stressors. Nutritional stress, food scarcity, or imbalanced diets can constrain growth rates, delay development, or alter morphological features in insects. Developmental plasticity enables insects to optimize resource allocation, prioritize survival or reproduction, and cope with variable or unpredictable environments. Interactions with microorganisms. Nutrition interacts with microorganisms such as symbiotic bacteria, fungi, and protozoa that inhabit the gut, cuticle, or reproductive organs of insects. Gut microbiota influence nutrient digestion, absorption, and metabolism, enhancing nutrient utilization efficiency and growth performance in insects. Microbial symbionts produce vitamins, enzymes, and secondary metabolites that complement the insect diet and support physiological functions. Dysbiosis or disruption of microbial communities can impair nutrient uptake, immune function, and growth in insects. In summary, nutrition plays a central role in the growth and development of insects, providing energy, structural components, hormonal signals, and regulatory cues for physiological processes such as molting, metamorphosis, and reproductive maturation. Understanding the interplay between nutrition and growth in insects is essential for elucidating their nutritional ecology, life history strategies, and ecological adaptations to diverse environments. Nutrition and development, ChatGPT. Nutrition plays a fundamental role in the development of insects, influencing various physiological processes, morphological changes, and life history traits from egg to adult. Here's how nutrition impacts development in insects. Embryonic development. Nutrition received by the female during egg development and oviposition can influence embryonic development and viability. Maternal provisioning of nutrients, including proteins, lipids, carbohydrates, vitamins, and minerals, in eggs provides essential resources for embryogenesis, organogenesis, and metabolic processes in developing embryos. Adequate nutrition promotes egg viability, embryonic growth, and developmental success, whereas nutritional deficiencies or imbalances can impair embryo viability, cause developmental abnormalities, or decrease hatching rates. Larval growth and molting. Larval growth and development in insects are heavily dependent on dietary nutrition, with larvae consuming large amounts of food to support rapid growth and tissue differentiation. Nutritional quality, quantity, and composition of the diet influence larval growth rates, body size, and developmental timing. Larvae undergo successive molts, shedding their old exoskeleton and growing a larger one to accommodate increasing body size. Molting requires energy, amino acids, chitin, minerals, and hormones, metamorphosis,